I say history of immunology, I won't be talking about the entire history, obviously that is not going to happen, it's going to take a lot of time. So briefly, what are the important aspects as far as immunology is concerned, okay? How this subject actually came into existence, that is what we are talking about. Okay, so immune in Latin means exempt. Okay, so if somebody is exempted from getting some disease, then we say that that person is immune to that particular disease. Okay, so that is the basic thing. Now, the first observation was made by it is pronounced Thucydides. Okay, he is a Athens historian. Okay, so during that time, plague was very, it was all over Europe. Okay, so many people suffered with plague, and the death rate was also very high. It is then that he observed that people who actually survived plague never developed plague or never got plague later on in their life. Okay, so they are exempted from getting plague once they are exposed. Okay, so that is how this historically is the first observation of immunity. Okay, though they didn't know the concepts, they didn't know anything, this was a major observation which was made by him, that once people get exposed to plague, and if at all they survive, they are not exposed, they're exempted from getting the plague again. Okay, this was the observation made. Now it took almost, this was in 5 BC. Okay, it took almost 2000 years before somebody else actually came up with a concept like this. Okay, so this is in 10th century, there's a process called variolation. Okay, variolation is a process where you are artificially inducing immunity into a person. Okay, so this is, you are inducing immunity. You're inducing immunity. Okay, so people from China and Turkish people, okay, Chinese and the Turkish people, what they used to do is, they used to, this was used for smallpox actually. Okay, what they used to do is, smallpox was dangerous back then. Okay, now it is almost eradicated, in fact completely eradicated. But back in 10th century and till most part of the history, you see smallpox being a big problem. Okay, so it usually ended up with death or several deformities. Okay, so what they used to do is, they used to collect the, smallpox, you get these pustules. Okay, so they used to collect the shells, okay, of the pustules after they get dried up. They used to make powder, either inject it under people who are, under the skin of the people who are healthy, okay, or they made them to inhale that powder. Okay, so the idea here is, people once exposed to smallpox, the same idea which the historian made, okay, the same idea, if you are exposed to smallpox once, then the probability of getting a smallpox later on in your life is absolutely zero. Okay, that was the idea. So they injected this powder directly under the skin, or they made people to inhale this powder. Okay, so most of them used to get a mild form of smallpox, and then they used to survive later on, and they never faced smallpox again in their life. And some used to die. There was no control here, and there was no, it's all a lottery basis. And some ended up with several deformities. Okay, so these were all the things, these were all the problems, but then the results were much better. Okay, rather than dying because of smallpox later on sometime in the life, people thought, okay, let's take some precaution. Even if we end up with a mild form of deformity, we'll survive later on in life. Okay, so that was the idea here. Now, the same idea later on was used by a lady from Istanbul, Lady Mary. Okay, her full name is Lady Mary Watley Montauk. Okay, so she is a lady from Istanbul. Yeah, she's a lady from Istanbul who actually, she had smallpox. Her brother also had smallpox, and unfortunately her brother died, but she survived. But she came to know from the European countries about this technique called variolation. Okay, so she administered just to make sure that her children didn't die because of this. She administered her children with the same thing. Okay, so that is how this variolation process became very popular. Okay, later the king himself asked her to come and demonstrate this in the King of Britain. She demonstrated that, and the king's kids, even they had the same process. Okay, so this is the first kind of vaccination that actually started around this is around 18th century. Okay, around 18th century, this is what was going on. Then Edward Jenner is another major contributor. Okay, we all know. Now, what did Edward Jenner do? Edward Jenner is the one who did the anthrax experiment. Okay, so what he did was, he took a cow which died of anthrax. Okay, collected the blood from the cow, injected into the ear of a rabbit, and then the very next day the rabbit actually died with anthrax. Okay, so he isolated the lymph node from that rabbit and then found the same bacteria which was initially present in the blood of cow. So that is how we established that the microorganisms, specifically in this case bacteria, are actually responsible for causing the infection. Okay, so he was the one who actually came up with the Cox. Sorry, I'm sorry. This is not Edward Jenner. I'm sorry. This is Robert Koch. Robert Koch, I'm sorry. So Robert Koch is the one who came up with the experiment, and then he later on proposed the Koch's postulates. Okay, later on he proposed the Koch's postulates. Now, before Robert Koch, around the same time in the 18th century itself, we have Edward Jenner. Now, Koch's postulate basically suggests, so he made a postulate saying that these criteria should be followed. We'll discuss the postulates later. These criteria, he made a four-point criteria saying that these criteria should be followed in order to establish that a particular organism is actually causative agent of a particular disease or an infection. Okay, so we'll look at the Koch's postulates later. Then Edward Jenner. What Edward Jenner did was, Edward Jenner did an observation. He made an observation. This is also in the 18th century. What he observed was, he had a maid who developed cowpox. Cowpox is similar to smallpox, but it is caused because of maids who actually milk the cows. So they are getting this a form of vaccinia virus. It's not vaccinia, it's a virus called the cowpox virus, which is similar to the vaccinia virus. Okay, this cowpox virus is similar to the vaccinia virus. Vaccinia virus is the one which causes the smallpox. Okay, so this cowpox virus is similar to that, and while milking, this virus gets transferred to the maid and people who actually work near the cows. Okay, so people who actually got the cowpox because of this virus, they did not develop smallpox, or they didn't have smallpox. Okay, so this was one observation made by Edward Jenner. So people who are exposed to cowpox never or rarely exposed to or got smallpox. So this was one observation made by him. So he wanted to test it out. So what he did was, he took a boy and then he injected cowpox virus. Okay, he injected cowpox virus into a boy. Okay, and then after a few days, okay, he then injected the smallpox, either the pustular solution or fluid or the powder from the pustules, okay, the shells. So basically he developed cowpox, but he did not get smallpox. So directly establishing that if somebody develops cowpox is a very mild form of smallpox. So it didn't have any side effects, like, even though the same thing was followed by Chinese, what they did was they used the smallpox shells directly. But unlike them, he used a mild variant of the smallpox, which is the cowpox, and then developed immunity, or he exempted the people from getting the smallpox by administering the cowpox virus. Okay, so this was the contribution of Edward Jenner. So basically he is the one who developed the concept of vaccination was developed by Edward Jenner. Then Robert Koch, to his credit, also has, he is the one who actually cultivated the bacteria in the lab. Okay, he was the one with the help of his assistant, he was the one, not assistant, his wife actually, who suggested using agar in the medium. Okay, so he was looking for some way to make a semi-solid medium for growing bacteria. Okay, then his wife suggested that why don't we use agar, the same thing which they used to use for soups, making soups. So that is how the concept of agar based cultures actually came in. So and together with Louis Pasteur, he actually came up with the germ theory. Okay. Then we'll look at his contributions. So he is considered to be the father of immunology. Okay, and what he did was he did the cholera bacillus experiment. So he was working with the chicken cholera bacteria. Okay, so one summer what he did was he left the flask and he went for vacation. Okay, so when he came back, he was actually supposed to inject this virus into the chicken to for his studies actually. So he left it, and then when he came back, whether he was lazy or he didn't want to waste the bacteria, whatever the reason was, he just checked the viability of the bacteria in the flask and then injected the same thing into chicken. To his surprise, none of the chicken actually died, none of them became ill. Okay, so that is when he thought, okay, why not try this? Then he injected the chicken with a virulent strain again. The same chicken which were initially injected with the bacillus which was sitting on the table for a couple of days, a couple of weeks actually. So they didn't, they were resistant, or they were immune to the virulent strain now. Okay, so that is when he understood that they're exhibiting the same concept of the what Edward Jenner has shown. Okay, so the same vaccination or the same variolation process which Edward Jenner actually developed. Okay, giving cowpox, the same thing. So what the explanation given by him is that if you leave the bacteria on the, or if you leave them long enough, they'll become attenuated. So he came up with the concept of attenuation. That is, they'll still be immunogenic, they'll be able to induce immunity, but they are not virulent anymore. Okay, they cannot cause the infection, but they can induce immunity. Okay, so when he left them on the table for a couple of weeks, they lost the virulence or the ability to cause infection, but they retained the ability to induce immune system. Okay, to stimulate immune system. So then he coined the term vaccination. Okay, because of the vaccinia virus which was first used by Edward Jenner. Okay, so just to give him credit for the process, he coined this term vaccination and called it a vaccine. So this is the first time that something like this have been reported. Then later on he actually developed the anthrax vaccine, and also the first human vaccine, that is the rabies vaccine. Okay, so he administered Joseph Mister, a boy who was bitten by a dog with this, and then he actually saved him, okay, from getting rabies. Then during this period, when people were actually unable to decide what are the factors which are actually responsible for the immunity, or which factors are actually providing the immunity, there were two groups of people actually. One led by Mechnikov, and one led by these were the major people. So Mechnikov believed he's a cellularist. What he observed was, okay, he actually observed in the lab that certain cells engulf other molecules, okay, other molecules and other smaller cells. Okay, so that is when he came up with the concept saying that the protection or immunity is actually contributed by cells. Okay, he said cells are the ones which are, because he did a practical experiment and he could actually see that certain cells are actually phagocytosing or they are showing phagocytosis. So he is a cellularist. Then Von Behring and Kitasato, what they were doing, they are they said it's not the cells, it is the fluids in the body that are actually responsible for the immunity. Okay, certain fluids in the body are actually responsible for immunity. So who do you think are right? Sorry, the cellularists? You have both. You have cellular immunity, which is contributed by certain cells, and you have humoral immunity, which is contributed by your complement, your antibodies, okay, everything. Okay, so both are right. Okay, so these people actually demonstrated. He demonstrated by phagocytosis, and these people demonstrated. This Von Behring is the one who actually developed the diphtheria toxin. Okay, for which she got the Nobel. Okay, so she demonstrated that you don't need the cells. So she collected the fluid from a person who was the blood from a person who was infected with infected and recovered from diphtheria, and then injected into, not a person, first animal experiments were done. Anyway, injected this antitoxin. Basically, they didn't know that antibodies were existent. Okay, they didn't have the concept of antibodies. So when the antitoxin was transferred into another animal, okay, that animal now becomes immune. So suggesting that some things in the blood are actually responsible for immunity. Okay, so these were experimentally proved, these were experimentally proved. Now we know that both the cells and the humoral components, both are important for the immune system. Both has to be there. Then next major contribution came from Paul Ehrlich. So he is the one who predicted, to his credit, he is the one who actually predicted the immune bodies. He didn't call them antibodies. So he said certain components in the blood are actually responsible for fighting with the antigens, okay, or the microorganisms, and providing immunity. And he even went further and said that certain hematopoietic cells actually have certain receptors. Certain hematopoietic cells have receptors. These are the receptors which actually interact with the microorganisms or the antigens. Okay, these receptors are responsible for interaction with The microorganisms are the antigens, and these receptors, upon interaction with these hematopoietic cells, these receptors are released. Okay, basically these cells start releasing the receptors continuously once they interact with the antigen. So the whole concept was put forward by Paul Ehrlich. Okay, so this, we know that everything that he predicted is perfectly true now. Okay, we know the concept, we know the clonal selection theory and everything. So based on this, again, the last, Burnett, he is the one who put forward the clonal selection theory. Okay, which basically says that when an antigen enters, it binds to or it interacts with a particular immune cell. Okay, in this case a B cell, now we know that. Okay, now once it interacts with that, it gets activated, the particular B cell, not the entire thing, not all the B cells. One particular B cell which is specifically selected by the antigen gets activated, it starts getting proliferated, it starts getting divided, it divides into your effector cells. We'll discuss that in detail, don't worry. So it effectively you have, so this is what Burnett proposed. So you have cells with basically receptors. Okay, when an antigen enters, the antigen will interact with a receptor which is compatible with, so you have several clones. So you have B cells with these receptors, then you have some B cells with this particular receptor. Okay, suppose if the antigen is like this, so obviously this will interact with the one which is circular, so compatibility will be there. Now once it interacts with this, only this clone will start to proliferate. Okay, so this will divide, you get two groups of cells, one is called the effector cells and the other one is called the memory cells. The effector cells will start secreting antibody, which is nothing but the receptor sitting on the B cell. Okay, so it will make more and more of those, but now instead of keeping it on its surface, it will start secreting them. And what do you call these cells? Plasma cells. Plasma cells. Okay, so you get two groups of cells, one is plasma cells and one memory cells. The plasma cells will start secreting the antibodies continuously while the memory cells sit there for a secondary response. In future, if you get exposed to the same antigen again, then instead of binding it again, dividing and then converting into effector and memory cells, simply the memory cells can directly go back to, plasma cells. Okay, that's the reason why the first time you get an infection, it will take a lot more time to recover than the second time, because you don't have to go through all the process. So you don't have to go through the activation of the particular B cell clone, then differentiation, then getting two different types of cells, and then the memory cells and the effector cells will now start producing the antibodies. The effector cells are nothing but the plasma cells. Instead, what do we have here? We already have memory cells, so our immune system has memory. So the memory cells are sitting there. If you are exposed to the same antigen again in the future, the memory cells will directly capture that, the memory cells will directly capture that, directly shift to plasma cells and then start secreting antibodies. That's the reason why the primary response will take a lot more time than the secondary response. Okay? Anyway, this is the clonal selection theory in short. We'll look into the details. So these are the major contributions as far as the history is concerned.